A relative new and upcoming technique within biomedical science is the growing of organoids. Organoids can be seen as microorgans with the same basic 3D structure and anatomy as a normal-sized organ. Organoids are grown from either embryonic stem cells, adult stem cells, or induced pluripotent stem cells. Stem cells are cells in our body that can form every cell type of every tissue in the human body. This is called pluripotency. Specializing of a stem cell into a certain cell type is called differentiating and cannot be done anymore by cells that are already specialized. Also, in contrast to specialized cells, stem cells can divide and form new stem cells indefinitely. In the first few days of a pregnancy, a blastocyst is formed. The blastocyst has an inner cell mass that consists of pluripotent stem cells, also called embryonic stem cells. When grown up, there are still some stem cells left in our body, adult stem cells. These stem cells are not pluripotent anymore as they cannot form every cell type in the human body. These adult stem cells can still form multiple cell types within a certain tissue and are somewhat capable of dividing into new stem cells. A third type of stem cell are induced pluripotent stem cells. These are stem cells that are developed from specialized cells. This is a relative new technique where growth factors are added to specialized adult cells to de-differentiate them into pluripotent stem cells. When wanting to grow organoids, the stem cells have to be manipulated into differentiating into the needed cell type. This can be done by adding growth factors to the stem cells. Growing organoids is a delicate job that requires adding the right growth factors at the right amount at the right time of development. Also, the environment of matrix in which the organoids are grown is crucial for the development of a 3D structure. Organoids of many organs have been made and used in studies. Examples are organoids of the brain, intestine, lung, and kidney. Organoids give us a lot of insight into the mechanisms by which organs and certain processes work. For example, brain organoids have provided the first direct human evidence for a causal connection between the Zika virus and the brain disorder microcephaly. Also, a boost is given towards the development of personalized medicine. Stem cells can be taken from a patient, organoids can be grown, and drugs can be tested on the microorgans of a patient to see the effects. Because organoids are a model for normal-sized organs, model animals don't have to be used as often in the lab. An issue which researchers have encountered is the lack of blood vessels in the organoids. Without blood supply, the cells in the center of the organoids don't get enough nutrients and die. This becomes increasingly a problem when the structures become larger and the nutrients can't be taken up from the environment. However, when the shortcomings are eliminated, there's great potential in this technique.